Hello everyone, welcome to the 7th student. Today we're doing the specimen paper, specifically theme C for higher level. So let's get started. Starting off with paper 1A, the first question is the diagram represents the metabolic pathway that converts threonine to isoleucine. What is a feature of this pathway? As always, I'm going to count down from 3. You can pause in the meantime to think, and then I'll say the answer and explain why. So 3, 2, and 1. It's D. How come? Okay, so it's not positive feedback. In fact, what happens here, okay, is that isoleucine inhibits threonine deaminase, right? So it's negative feedback. Isoleucine is not a competitive inhibitor. It binds to the allosteric site of threonine deaminase, so it's non-competitive. And then isoleucine is a substrate for threonine deaminase. It's not a substrate, it's an inhibitor, right? There's a difference there. The substrate is the thing that is catalyzed by the reaction. So isoleucine is an allosteric inhibitor though, because as I said, it's non-competitive. That way, this pathway is regulated. When there's too much isoleucine, it inhibits the enzyme, which in turn inhibits the pathway. Great. Next question. What's a common feature of enzymes? I think this one's pretty straightforward. 3, 2, and 1. It's A, right? So they all react with substrates. That's the whole point of an enzyme, right? They catalyze the reaction from substrate to product. They increase the rate of reaction. They're not all secreted. Some stay inside the cell, right? Remember, the free ribosome ones stay inside the cell. The ER ribosomes leave the cell. And they all bind to the active site of their substrate. It's the other way around, right? Next question. The diagram shows the movement of ions that can occur across the membrane of a neuron. From the diagram, what can be deduced about the movement of sodium ions? Once again, 3, 2, and 1. Right. So sodium is actively pumped out by the sodium-potassium pump, as you can see here. So 3 sodiums go out and 2 potassiums go in. And then it goes back through facilitated diffusion. So this is just going through a channel. There's no energy expenditure here, whereas the sodium-potassium pump uses ATP, as the diagram shows. I think that's also pretty straightforward. As always, any questions in the comments, please. Okay, so what activates the tyrosine kinase pathway in a cell following binding to a receptor? 3, 2, and 1. Insulin, okay? If you don't remember this, go back to that topic, chemical signaling. There's a whole section on this, right? But remember, when insulin binds, 2 tyrosine kinases on the intracellular side of the receptor phosphorylate each other, and that then triggers the pathway. But again, you can check that out in the video. So on to the next question. What is the cause of positive phototropism? So 3, 2, and 1. The next question is, which role does positive feedback play in fruit ripening? And once again, 3, 2, and 1. Well, before we look at the answer, I want to quickly mention that all of the slides for all the themes, including extensive notes for each, are all available on my website now. And if you're not convinced, you can download a sample to decide whether they're worth it. But moving on, the answer is B. How come? So when a fruit starts ripening, right, it's because ethylene is causing it to ripen. But then when the fruit ripens, that causes the release of more ethylene, right, which causes the fruit to ripen, which causes the release of more ethylene. So it's a positive feedback loop, right? It self-amplifies. That's why it is called positive feedback. So next question, how is involuntary peristalsis in the intestine directly controlled in humans? So again, 3, 2, and 1. It's the ENS. Okay, so remember, swallowing and defecating is the central nervous system. Those are the only 2 things involved with the gastrointestinal system that are conscious. But then the enteric nervous system controls peristalsis, right, which is involuntary. Great. Next question, which cell is a component of the innate immune system? This one's pretty straightforward. 3, 2, and 1. A phagocyte, right? Remember, phagocytes engulf pathogens, right? Again, you can check out the video for the immune system, but then all the other cells are adaptive. So those learn throughout time and they're involved in the secretion of antibodies. So quite different. Next question, which is a density-independent limiting factor for a kangaroo? So once again, 3, 2, and 1. Okay, the first time I read this question, I have to admit, I read it incorrectly as density dependent, but it's important, it's density independent, meaning predation is a typical example of a density dependent factor, right? The more prey, the more predation, right? Because there's more available. Whereas a forest fire is completely independent, right? It just is going to kill everything in the forest. It doesn't matter if there's more foxes or less, for example, right? It's just going to kill all of the foxes in the forest. Eutrophication is not a density independent or dependent limiting factor. It doesn't control the growth of a population. I mean, it can through reducing the oxygen available, but it's not considered one of these factors. And same thing with climate change. So now we move on to paper 1B. So this first question is super long. I'm not going to bother reading the entire thing, but again, pause now, read the whole thing, and I'm going to read the actual question, which is outline the concept of mutualism with respect to this example. And again, in 3, 2, and 1. All right, so there's clearly a symbiotic relationship between two unrelated species, right? The Rhizobium and the Fabaceae. This is again in the syllabus, so you can go back and watch that video. But the bacteria get organic molecules from the plant, from photosynthesis, and the plant gets nitrates, which are often very hard to get for plants. So the next part is distinguish between the cells in Rhizobium and Fabaceae. So 3, 2, and 1. So Rhizobium, right, they're telling us it's a bacteria, so it's a prokaryote, whereas Fabaceae is a plant, so it's a eukaryote. Those are two main things, right? And then you can just start listing differences between prokaryotes and eukaryotes. So prokaryotes have circular DNA, eukaryotes have linear DNA, right? Prokaryotes do not have organelles, eukaryotes do. Prokaryotes have 70S ribosomes, eukaryotes have 80S, right? And prokaryotes are smaller and unicellular, right? Whereas eukaryotes, in this case, a plant, is multicellular. Great. The next question is, suggest a reason that mutualistic relationships often involve species from distantly related groups of organisms. So have a think. I think this is somewhat intuitive. You don't really need to remember this. In 3, 2, and 1. So in a mutualistic relationship, right, both are giving something that the other cannot have by itself. So the more distantly related those organisms are, the more likely it is, right, that they have different characteristics. That's needed, because if they're the same, then they don't need each other, right? So you need something that the other doesn't have and that provides for the other's requirements. So next question, analyze the results of the experiment. So 3 marks, so take your time, but 3, 2, and 1. All right, so obviously, when you increase the nitrate, the nodule size does not change, right? That's this line here. Without additional nitrate, though, it does increase. So nitrate is obviously inhibiting nodule growth. When nitrate is scarce, nodule growth ensures nitrate supply from a larger bacterial population, right? This is what we were talking about. So the nodules, right, have the Rhizobium on them, which provide the nitrate. And this suggests that mutualism is affected by environmental conditions, right? Depending on whether there's nitrate or not. If there's a lot of nitrate, you don't need the Rhizobium, right? So you don't need to grow these nodules with the bacteria on them. You can provide that for yourself. But if there's very little nitrate in the environment, in the soil, then you're going to need them to provide it for you. Great. So now the last part, which is paper 2. This question shows the movement of auxin across the plasma membrane of a plant cell, as well as the position of auxin efflux carriers. So first, it's asking you to annotate the diagram with one arrow to show the direction of the net flow of auxin through this cell. Think. And in 3, 2, and 1, it's going to go this way. How come? Okay, so auxin is coming into the cells, right, through diffusion. And then all of the carriers that you can see here, right, are facing this way. So this suggests that there's a directional flow, right, from the left side to the right side. It's going to enter the cells and then leave through these carriers. Because remember, auxin cannot leave a cell by itself. It needs the carriers, right? And again, that's in chemical signaling. You can go take a look. So if that's clear, the next question is identify an environmental factor that causes auxin efflux carriers to be concentrated on one side of the cell. And again, 3, 2, and 1. Gravity or light, right? This is what tropisms consist of. So if suddenly the light is found here on this side, right, the plant's going to want to have the auxin on the other side, on the shaded side. So it's going to put all of the carriers on this side so that auxin flows in this direction. So moving on, predict the difference that could be seen using a microscope between plant meristemic cells grown in media with and without auxin. Pretty straightforward again. 3, 2, and 1. Obviously, if there's auxin, they're going to be larger, right? That's what auxin does. Remember, it increases the concentration of proton pumps on the membrane, so that then the pH is lowered on the cell wall, basically, right? Because protons flow in, and that causes the cell wall to be weaker, so it's able to be broken and enlarged. It's a little bit of a complex concept, but it's cytokinins, right? So cytokinins, remember, interact. They can be synergistic, so meaning they both contribute to growth, or antagonistic, where one contributes to growth and the other inhibits growth. So they work together. Next question. Predation may result in a cyclic pattern of population changes. So, for example, the lynx and the hare. The predator benefits from a predator-prey relationship by obtaining food. Suggest how the hare population can benefit from this relationship. Now, this is something you've probably never thought of, but 3, 2, and 1. Okay, so it doesn't actually help an individual hare, right? But as a population, it ensures that the habitat is sustainable, right? As a population, it removes the genetically weaker individuals. As a population, it prevents overgrazing. Now, I know for humans this is quite difficult to understand because we see human loss as a tragedy, but you have to understand that in a natural ecosystem, right, it's very rough. There's a lot of competition for resources. So having less numbers or sustainable numbers, right, means that they can all live and have resources instead of having to fight to eat and reproduce, etc. So as a population, it can be beneficial to have predators. So the size of the predator population, that's a really big one. Obviously, the more predators, right, the less prey because they're being eaten. Also the resources and food because the prey has to compete for that, right, if there's a limited amount. And also pathogens. So that's another density dependent factor because pathogens spread best when there's very, very dense populations, right? You could see that during COVID in cities it spread way more than in the countryside because we were more densely packed. So that is the third example of a density dependent factor limiting carrying capacity. Great. Now it asks to outline how the hare population size can be estimated using the mark release recapture method. Once again, this is something that's easy to remember. It's C4.1. But 3, 2, and 1. So, okay, it seems stupid, right? But you just have to define what it is. You capture a sample, you mark it. You can give more detail here, right? You try and mark it with something that's not very obvious, then you release it, and then you recapture another sample. And then you can use the Lincoln index, right, to calculate the size of the population. There's a lot of assumptions here, which is actually the next question, so I'm not going to spoil it. So the next question is state one assumption that's made when using this method to estimate population size. And so now, hopefully you remember one. 3, 2, and 1. So all of these are assumptions that you need to use that method, right? A good example, I think, is just low levels of immigration and emigration, right? Meaning no new members of the population enter or leave. Great. And then the final questions that we have to do are the long answer questions. So there's quite a few for theme C. I think there's 3 or maybe even 4. So explain how vaccination can result in specific immunity to a viral disease. Now take at least 4 to 5 minutes to think about this. So pause now, outline your result, and in 3, 2, and 1, the mark scheme goes as follows. So remember, vaccinations, right, are specific to a disease, right? How come? Because they trigger the adaptive system. So remember, vaccines are going to have an attenuated version of the virus or an antigen or nucleic acids of the virus, right? Or a dead version of the virus. And this basically is going to trigger an immune response, right? The antigens are going to be recognized. Remember, the phagocytes are going to try and eat it. Then the lymphocytes, so the T cells, are going to get activated. The B cells will get activated by the T cells then, and the B cells will produce antibodies. They'll divide into plasma cells, which will provide a quick response, and then they'll divide into memory cells. And those memory cells are the key ones because they provide long-term immunity. That's how vaccination works, right? It triggers these memory cells, which then, if you get the pathogen again, the disease, you get a much, much quicker response. So all of these things are really important to mention. I think you can order it how I talked through it, right? Just from start to finish, like what happens first, the innate, then the adaptive, and how that leads to memory cells. That's a pretty good answer, I think. So next question. Outline the role of membrane proteins in the movement of specific ions at specific times in the transmission of nerve impulses. So hopefully you remember which direction everything flows in. Once again, take a couple minutes and in 3, 2, and 1. Okay, so you need to remember what happens at every stage, right? During depolarization, the sodium channels open, so they flow into the axon, then repolarization, the potassium flows out of the axon. That's all facilitated diffusion. And then the sodium-potassium pump uses active transport, right, to get the membrane back to resting potential. So it moves all the sodium that moved in outside, and it moves all of the potassium that moved outside inside. And it does so through moving 3 sodiums out and 2 potassiums in. So that's important to remember. I'll link to the topic in case you want to have a quick recap. Now, moving on to the penultimate question. This is a 2-part question. So the first one is, toxins often act as inhibitors. Compare and contrast competitive and non-competitive enzyme inhibition for 7 marks. So again, take a good 5 minutes to 10 minutes to outline this answer. And in 3, 2, and 1, okay, so you have to pull out all the stops here, right? You need to remember everything. So obviously, you need to give similarities and differences because it's compare and contrast. So similarities, they both slow down enzyme activity, right? They both interact with R chains of amino acids at the surface. They both bind to the enzyme. That's important, right? Now, differences, whereas competitive binds to the active site, non-competitive binds to the allosteric site, right? That's why in competitive, you can outcompete the inhibitor if you're a substrate, but in non-competitive, you can't bind to the allosteric site, so you can't outcompete it. So that's the second part, right? So it stops the substrate from binding. The other one just does not stop the substrate from binding, but rather causes a change in the active site. So then you can say that here as well, right? Competitive is reversible. Non-competitive is normally permanent because you can't take it out of the allosteric site once it's in it. The competitive inhibitor has a similar shape to the substrate because it's binding to the same site, the active site, whereas the non-competitive is binding to a completely different site, so it's very different. And competitive can be reversed by increasing substrate concentration. That's true, right? As I mentioned, you can outcompete the inhibitor with more substrate, whereas in non-competitive, you cannot, because the substrate is still able to bind to the active site semi, right? But it just doesn't work because the active site has been changed by the inhibitor binding to the allosteric site. So those are all the key points to hit, right? But be sure to include a similarity. I think that's really important. Just make sure that you include both act as inhibitors and define what an inhibitor does in the first place. Great. And then the last question is, rotenone is a naturally occurring toxin that blocks the electron transport chain in insects and fish. Outline the consequences of exposure to a toxin like rotenone for cell respiration. 4 marks. So here you basically just have to tell me what the electron transport chain does, right? And what's going to happen if we block it. So say the opposite of everything that normally happens, right? This will not be able to happen. That will not be able to happen. So think, and in 3, 2, and 1. Okay. So obviously the ETC is involved in aerobic respiration. So you're going to have less aerobic respiration. Also remember that in the ETC, we turn NADH into NAD, right? So if we can't do that because it's blocked by a toxin, then we're going to get NADH accumulating or reduced NAD, right? It's a synonym. Also, we're going to get fewer electrons to be transported by carriers. What's going to happen? So if it's blocked, the ETC, the electrons are going to accumulate, and over time there's not going to be any more space for electrons, meaning they're all just going to accumulate in NADH and FADH. Also, if we can't move electrons through the carriers, we can't transfer protons, right? Because that requires that energy. So we're not going to be able to maintain a gradient. And therefore, also no protons are going to go through the ATP synthase because there's going to be no gradient. Therefore, ATP is not going to be created. And finally, right, oxygen, which normally accepts the electrons at the end of the chain, is not going to be able to do anything because there's not going to be any electrons flowing through. So you see the idea here, right? It's just giving everything in the negative. So I know this topic is quite complicated. Any questions, leave them in the comments. And until then, I'll see you next week.